So we have seen in detail about secondary metabolites. What are they? What kind of compounds they are? They are produced by microorganisms as well as plants. They are not actually useful in any way to the organisms, but their commercial value can be exploited based on the kind of the metabolite. So some amount of metabolite may be of some use to the living organisms, but most often 99% of the secondary metabolites are exploited commercially for the benefit of the humans and the animals only, because we are not able to produce any secondary metabolites. So plant secondary metabolites, yeah, we are dealing with only plants, but you also have microbial secondary metabolites. In fact, microbial secondary metabolites are more, what to say, they are more vigorous than the plant secondary metabolites. So they are normally plants produce secondary metabolites mainly for their defense. Most often they produce it for their defense only, to defend the pathological conditions caused by microorganisms or any other competing plants, parasitic plants, or any competing plants competing for nutrition, for a particular nutrition, or for some, mostly for nutrition and space. So a particular plant will produce some germination inhibiting substance, so it actually kills the competing plant, or it inhibits the growth of the competing plants, or any kind of herbivores against insects, against any kind of. Adathoda is one typical example for secondary metabolite production against defense by a herbivore. So goat, ஆடு வந்து அந்த பிளான்ட் தொடவே தொடாது. So common name adathoda has come only from that. So it produces certain alkaloids which inhibits it to be eaten by any herbivore. So it causes a repellence, deterrence, toxicity, growth inhibition. So இதெல்லாம் வந்து these are the conditions by which the secondary metabolite defends the plant, okay, by exhibiting toxicity against the microbial pathogens, by killing the pathogens or inhibiting the growth of the pathogens. So this is the most common application of plant secondary metabolites, why it is being produced in the plants, how it is useful to the plant. It is not useful for the growth of the plant, but it is mainly used for defending the plants against diseases or any kind of stress induced in the plant. And on the other hand, secondary metabolites are also used for attracting pollinators. So some volatile oils are being produced, like for example citrus plants, they often give out volatile essential oils through their leaves and stems and flowers. So there are many secondary metabolites. All the odorants, no, the flower, the flower smells very good. Some flowers are very good smelling flowers. So this smell itself is a ejection of excretion of volatile chemicals. Okay, so these are secondary metabolites only. So sweet scent which emanates from the flowers are nothing but secondary metabolites produced by the plants. So some pollinators get attracted by the scent. Some pollinators get attracted by the color. The color or the pigment produced by the flower is also a secondary metabolite. So the color is produced to attract certain group of pollinators, like butterflies. They are generally attracted to yellow colors. Honeybees are generally attracted to some other color, or honeybees are generally attracted to scented flowers. So like that, each flower or plant produces secondary metabolites apart from defense mechanism for pollination also. So it can be any secondary metabolite which leads to good smell or scent, which leads to bright colors, okay, which gives attraction, which attracts the insects or the animals, which attracts seed dispersing animals. Okay, one more very good example is root nodule bacteria. How the nodule is formed in the roots of the leguminous plants. So rhizobium is a soil dwelling bacteria. The legume roots excrete a secondary metabolite into the soil. So this secondary metabolite will attract the population of rhizobia. See, as soon as the legume is sown in the soil, what will happen? Whatever chemicals get extracted from the root will attract the rhizobial cells from the soil. Okay, so all the rhizobial cell will get attracted to the root and they will come and colonize the rhizosphere region, the soil immediately surrounding the root region. So when that is the first step of root nodule formation. So rhizobia, which are present in very small numbers in the soil, as soon as they get attracted by the excretion of the secondary metabolite from the root hairs, they will all come chemoattraction. This is called chemoattraction. So they'll all come and gather together surrounding the root hair. So this is the first step. After that, they produce the microbes also produce some chemicals, and then root nodule formation. That is a different story. But then uh, production of root nodule is depending on the release of secondary metabolites from the specific plant. So each legume is specific to its rhizobial population. So green peas will excrete some other metabolite to attract rhizobium. They madri beans, French beans will excrete some other metabolite. What is the Arachis hypogaea, groundnut will excrete different metabolite. So each one is specific to the plant also. So you can see how the secondary metabolites helps the plants in different ways, not necessarily for its growth, but in different ways, which is enhancing the potential of the plant. So we are able to exploit the production of secondary metabolite by from the previous days. They used to use the whole plant. They will need a lot of plants to extract the metabolite. But nowadays, when plant tissue culture has come up in a great way, we can get the required secondary metabolite using the callus culture or suspension culture, using just a little bit of cell mass, some grams. Gram connector cell mass is enough to produce the secondary metabolites continuously. You can also go in for continuous culture or immobilized culture. So that is the importance of the secondary metabolite production by the plants. So we have high value substance commercially high value substances. They have been produced using plant tissue culture methods like growth regulators, auxins, cytokinins, gibberellins have been produced. Hormones, insecticides, virus inhibitors are being produced. Biochemicals like amino acids, enzymes, lipids, nucleic acid derivatives, all vitamins, they are also coming under secondary metabolite. So they will enhance the amount of vitamins in the plants. You have certain secondary metabolites that enhances the amount of vitamins in the plants. Main thing is food additives and fragrances, because you have so many uh, sweet scented flowers, fragrant flowers. So main thing, they are used, they come under the group of flavonoids, okay, and terpenoids. All these sweet scented chemicals, they come under the compound of flavonoids and terpenoids. So food additives and fragrances, you have condiments, spices, fragrance, and the cumin seeds, the what is it, pattai lavanga flavor. Okay, that flavor you can get. Emulsifiers, flavors, okay, vanilla flavor, pista flavors, whatever, chocolate flavors. So what different kinds of flavors can be produced by the plants, they can be used commercially to in tissue culture methods. Fragrances, definitely they come a long way actually. Perfumes, definitely. Spices, okay. Sweeteners, you have artificial sweeteners. Stevia plant has been exploited commercially for producing artificial sweeteners. So again, medicinals and drugs la, Alkaloids are the most popular one because most of them are anti-cancer agents, allergens, anti-cancer agents, antimicrobial agents are there, cardioactive substances are there. You have this vincristine. Okay, then opiates are there, which are actually morphine group sedatives. Opiates are used as antidepressants. Now you have heard a lot about antidepressants. The people who have bipolar disorders, who have problems of depression, they are given opiate drugs, which are also extracted from plants. Then immunochemicals, you have immunosuppressors and immunostimulants. Immunostimulants as well as immunosuppressors can be used and can be extracted from the plant. And steroids are always there. Anti-inflammatory agents, you have they have most used in the clinical side to as painkillers and anti-inflammatory agents to reduce inflammation, to reduce swelling. Okay, they use steroids. So these are some high value substances. Apart from this, you have low value substances also, which do not have much commercial value, but still they are produced, they are extracted from the plants. We are going to only focus on few high value substances. So in this case, they have seen that instead of using the whole plant itself for extracting, cell cultures, they have seen the yield has come very high. Very To quote few examples, berberine is a alkaloid which is produced by Coptis japonica from Coptis japonica. 5% of dry weight after 5 years of root growth, if you take from the plant as such, you can get only 0.17 mg per gram. But in this case, you have 44 mg per gram per week if you use cell lines. So you have 250 times higher extracting capacity or production yield, if you call, from cell cultures when compared to the whole plant. That's why they have shown in research, these are the some of the secondary metabolites, high value secondary metabolites. These are the plant species, and you get cell culture yield which is shown to be definitely higher. Okay, definitely higher than the whole plant. Okay, so this column shows how suspension culture, callus culture. So S represents suspension culture and C represents callus culture. This also you can see suspension culture becomes is a very what to say, it is a chosen culture method because it gives a higher yield over the callus culture. So chiefly there are three ways for the production of secondary metabolites. We have seen many techniques, but for the commercial production of secondary metabolites, they use three methods: cell suspension culture, hairy root culture, and immobilized cell culture. Even though callus culture is available for higher yield commercial exploitation, they use cell suspension, hairy root, and immobilized cell culture. So root cultures, they have seen of the three what we have seen in the last slide, the root culture is better than cell suspension cultures. So instead of culturing the whole cell as suspension, they use root cultures. Okay, so what they say is the roots are better excretory organs for these metabolites into the surrounding medium, so it is easy for extraction and purification of the compound. And then they add additionally charcoal, by which they can absorb all the metabolites. It's easy recovery of the metabolites from the medium. They add charcoal because activated charcoal is a very good adsorption medium. It absorbs all the metabolites, get adsorbed on the charcoal, and then it is easy to remove it from the charcoal also. So this they have also seen through research that charcoal enhances the production of the metabolite also. So all these are research based evidence based studies only. So another important compound in this kind of commercial exploitation for production of secondary metabolite is elicitors. So elicitors are compounds of biological origin, which means you can get it from microorganisms or from any other plant. but only microorganisms offer low cost elicitors. Okay, if you use higher plants, it is difficult to extract the elicitors and then test for the production, for the enhancement of the secondary metabolite. So mostly they prefer microorganisms. It can be bacteria or it can be fungus, and it can be actinomycetes also. They are also very good secondary metabolite producing organisms, actinomycete. So they are compounds of biological origin which stimulate the production of secondary metabolite. So elicitation, enhancement, stimulation, all these are there are compounds. It can be from ions. It can calcium ions can elicit a particular secondary metabolite. Ion can select. It can be anything which is produced by any kind of compound produced by a living organism, a biological organism. So it can be a crude elicitor, or you can it can be a mixture of elicitors, or it can be a single elicitor. All these has to be optimized when you are doing research on secondary metabolite production. So it normally a mixture of elicitors are better for improvement of secondary metabolite production, instead of narrowing it down to one, because extracting one elicitor and applying it for enhancement is more cost oriented. It is more expensive. So what they do, they just grow some, they just collect crude elicitors and they apply it on this kind of medium. They add it to the medium and look for the enhancement of the secondary metabolites. So they are very production is very low, demand is not met. That means they have elicitors have not, in the production area they have not met the demand. Effort is for production for product formation at molecular level. They have exploited for increased production. So elicitors, you can have endogenous elicitors, exogenous elicitors, biotic elicitors. Few abiotic elicitors are also there. So endogenous, as the name suggests, within the cell. Exogenous, produced by microbes. See, plant cell itself can produce certain elicitors. The cell suspension culture which they are using, those cells can itself produce certain elicitors. Okay, so the pectin can act as an elicitor. Cellulose can act as an elicitor. Carbohydrate, the glucose which you apply to the medium can act as an elicitor, or glucose which is produced by the plant itself can act as an elicitor. So you have many endogenous elicitors. Exogenous elicitors is what we apply in the medium, we supply in the medium, like chitin, chitosan, glucans. It can be anything. It can be any ions, it can be heavy metals, it can be any compound, chemical compound which is produced organically in a living organism. Biotic, all elicitors of biological origin. This endogenous, exogenous, everything comes under biotic. And abiotic is UV radiation, cold treatment, subjecting the cell suspension to cold temperature, to subjecting the cell suspension to a higher temperature. It can be a fungicide, it can be an antibiotic, it can be many other chemical substances which are of non-biological origin. Okay, and as well as physical agents like osmotic pressure. You increase the osmotic pressure, look for the production of secondary metabolite enhancement. So all these come under abiotic elicitors. So all these have to be optimized for the final scale up study. Okay, so you can, another important compound you have to remember is precursor. Suppose you want to produce a secondary metabolite B, you have to add the precursor to the metabolite. Okay, so you have many precursors are available for production of any kind of substance. You have many precursors because the plant naturally produces the secondary metabolite in its metabolic pathway. So every metabolite excreted out will have a precursor. All these are enzyme mediated reactions. So from A to B, B to C, C to D, D is the final product which is excreted by the plant. So it has C is the precursor, immediate precursor of D. So if you identify what is the precursor, you can add the precursor in the cell suspension medium, culture medium. So the without the formation of A and B, directly D is produced if you add C to the medium. So the plant will already have the enzyme in the cell suspension. So it will produce the enzyme. Okay, so it will transform C into D using the enzyme. In some cases, they add the enzyme also from outside. Some cases they add the enzyme also, because if A and B are not produced, the enzymes also will not be there. So they will add only the previous compound to the final, that is precursor solvent. So if D is the final compound, they will add C to the medium, and they will add the enzyme also to the medium. So it is like that. So see, tropic acid is a precursor of tropane alkaloids which are produced by the datura. Succinyl benzoic acid is a precursor for anthraquinone, which is produced by cultures of morinda. They have seen, see, even if you don't add the precursor, the compound will be produced, because the cell has all the machinery for producing the compound, because it is a natural process, metabolic process. So even if you don't add precursor, the compound will be produced. But they have seen, if you add the precursor, the yield will improve. Okay, that is the main idea of commercial exploitation of the secondary metabolite. So addition of phenylalanine to the culture of Taxus capsidata, they potentiate and enhanced the level of rosmarinic acid and Taxol. Taxol is a very, very powerful anti-cancer agent. So this is commercially exploited by this plant, from this plant culture. So if you add phenylalanine, they get enhanced yield of this particular compound. Again, tryptophan is a starting material for indole alkaloids. Okay, all indole alkaloids, you need tryptophan as the starting material. It is not that the plant does not produce tryptophan. It produces tryptophan. It produces small levels of indole alkaloids. But if you are going to get higher amount, if you are going to need higher amount of the alkaloid, you need to add the precursor. In addition, you can add an elicitor which is not having any role in the metabolic pathway of the secondary metabolite production, but in some manner it boosts the production of the secondary metabolite. So that is the role of the elicitor. It has no direct connection with the metabolic pathway of secondary metabolite. So effect of precursors, as I already told you, these are all the precursors for the specific secondary metabolites. All amino acids, okay, pyruvates, amines, all these are precursors. Some example of precursors. So as I already told you, people have used a particular bacterium called Agrobacterium rhizogenes to form root cultures of different plants. So this particular bacterium is a causative agent for hairy root disease, just like Agrobacterium tumefaciens produces galls. You have studied tumors or gall diseases in fruit trees, okay, in roses, you can see in apple trees. Gall disease by Agrobacterium tumefaciens produces a big tumor around the stem region, above ground parts. Okay. Similarly, Agrobacterium rhizogenes, it also involves the production of a tumor kind of disease in the roots. So rhizonale, it is root. So it produces what is called hairy root disease. So when this is actually a pathogen. It is actually a plant pathogen. So scientists have used this pathogen and used it to produce a condition, cell culture condition, in which the cells are stimulated to produce secondary metabolites. So because this organism induces the formation of tumors. What are tumors? Tumors are nothing but unorganized mass of cells. So you don't have to spend too much time and money to produce a bunch of a mass of cells. So you have to use the explant, you have to wait for 4 weeks for callus to develop, and then callus to grow, you have to take another 4 weeks. It is very simple. If you infect the explant with Agrobacterium rhizogenes, within a week, you will get a tumor like growth. You will get high amount of cell cellular mass, which is the first requirement for production of secondary metabolites. For any cell suspension culture or a cell immobilized cell suspension culture, you need biomass, you need the cellular mass. The cellular mass is easily provided when you infect the explant by the hairy root causing pathogen, Agrobacterium rhizogenes. So cells are transformed. Actually, this has a plasmid, root inducing plasmid. It is called RI plasmid. You would have studied about Agrobacterium tumefaciens, TI plasmid, tumor inducing plasmid. So in this Agrobacterium rhizogenes has a large plasmid called RI plasmid, root inducing plasmid, and this plasmid can be attached to the uh, DNA of the cells. Okay, so that's how the cells become, they multiply very fast. So when the RI plasmid is attached to the integrated into the genome of the cell through genetic engineering methodology, the cell starts dividing on its own. Normally, if you use an explant for callus to develop, okay, or if a callus has to develop further, you have to add auxin cytokinin in a particular ratio. So medium la, when you place explant on the medium should contain auxin cytokinin ratio at a particular uniform level. Only then callus development will take place. After that only you go in for organogenesis, you increase auxin level, decrease cytokinin level for root shoot formation and all that. But before, you need to add a certain proportion of auxin cytokinin for development of callus. So in this case, the cells explant which is infected with Agrobacterium rhizogenes, you do not have to add any hormone, because this gene, this RI gene itself has, it has the capacity to induce production of auxin cytokinin by the cell itself. So the cell itself produces the required amount of hormones at the required concentration. That is how it divides very fast. It divides very fast into a mass of culture cell. So they're transformed with some of the bacteria's DNA, that is the RI plasmid, causes the cells to be more sensitive to the hormones they produce. So they themselves start producing the hormone. The cells form into roots. That is root inducing hormone. Okay, these roots grow very fast, produce the secondary metabolites that ordinary roots produce. So this is a very important mechanism, hairy root culture. So plant tissue culture, to produce secondary metabolites, you have many kinds of secondary metabolites. I have already told you. So culture is produced after the infection of the explants by a gram-negative soil bacterium, Agrobacterium rhizogenes, which contains RI plasmid. So they can infect the plant roots, cause them to produce a food source for the bacterium. Actually, plant roots which are infected by RI plasmid, they generally produce opines, which are the food for the bacterium. So this interaction between the cell and the bacterium allows the cell to multiply very fast. Okay, abnormal growth. So explants are wounded. They are inoculated with Agrobacterium rhizogenes. Wounded in means scalpel which just light scratch panvanga. They'll just wound the explant. After 2-3 days, it can be transferred to a solid media. You have to add some amount of antibiotics, because after 2-3 days you don't need this bacterium. You should not allow the bacterium also to grow along with the culture. So in 2-3 days time, the bacterium's RI plasmid gets inside the cells of the plant cells. So after that, you want to eliminate the bacterium, so you add antibiotics in the medium which kills the bacteria, which are no longer needed, because in 2-3 days time the RI plasmid gets transferred to the explant through the wound. Okay, so hairy roots will be induced within a short period of time, 1 week to over a month, depending on the plant species. So as short as 1 week is enough for producing the hairy roots. So decontaminated hairy roots culture can be subcultured on phytohormone free medium. You can see it is again cost effective. You don't need to add any hormonal mixtures into the medium. So this is how it looks like. First day, within a week you can see hairy root. This is root inducing hormone actually. So they produce high bunches of roots, and it is seen that these roots yield more secondary metabolites than normal roots. See, most of the plant, I have already told you, root culture is very, very, it is very good for producing secondary metabolites. So, normal roots vida, these hairy roots of the same plant can produce a higher amount of secondary metabolites. That's why they use this hairy root culture method. Okay. So advantages of hairy root culture is because they are fast growing, low doubling time. In very short time, they double. Okay. Genetic and biochemical stability. This is the most important advantage of when you want to commercially produce secondary metabolites, because you don't need, you don't it is not good for unstable culture lines, because it will affect the quality and quantity of the secondary metabolites. So genetic and biochemical stability is very, very important advantage that is available with the hairy root culture. And definitely growth in hormone free media, you don't have to add any hormone in the medium, so they can be used as continuous source for production of secondary metabolites. See, it makes things very, very easy, because you don't need you need machines only for purification and extracting. For production, this is the most cheapest way of producing secondary metabolites. So once you produce the metabolite from the medium, next step is extraction and purification. That you need laboratory vessels and bioreactors by which you can separate the metabolites. For production, it is enough if you use hairy root culture. So I will just take one: food additive and flavor agent. So you have natural colors and natural flavors. Okay, saffron is a very common. Anthocyanin, carotenoids, chlorophyll, curcumin, iron oxides, riboflavin, all these are natural colors. Okay, sweet potatoes, spinach, tomatoes. Okay, all these are antioxidants normally. So I'm just going to take one: shikonin. I hope you remember, this was first used, commercialized by Japanese for the production of bio lipstick. Okay, shikonin, the first